My name is Ulrich Diernagel of the Quest Center for Responsible Research at the BIH, and I welcome you and say good morning, at least to California, where it's 8 a.m. And that is the reason why we had to move the BIH lecture into the Berlin afternoon, where we have a full house. Okay, so now for today's talk and speaker, Dr. Anita Bandrowski of the University of California at San Diego, at the Department of Neuroscience, and she's also the founder of SciCrunch, of which she is also the CEO, and we're very happy that she is currently also a BIH visiting fellow of the Stiftung Charité. In her talk, Is the Scientific Literature Better Today Than It Was 10 Years Ago? She will explore two main topics: RRIDs, that's Research Resource Identifiers, these are persistent unique identifiers for key biological reagents, and the RTI, the Rigor and Transparency Index. Anita trained as a bench neurophysiologist, where she was working to elucidate physiological mechanisms of learning and epilepsy. But soon after her postdoc, she became a pioneer in several now highly successful international activities, locating, accessing, and utilizing research resources. Notably, she was involved in creating the Neuroscience Information Framework, an initiative of the NIH Blueprint for Neuroscience. The framework now is the most comprehensive search system for neuroscience data on the web. To address reagent biological underreporting issues, Anita founded the Research Resource Identification, or RRID initiative, which she leads. We will hear more about RRIDs in her talk. Anita is also the co-founder of SciCrunch and also its CEO, a company that helps diverse communities to increase the discoverability, accessibility, visibility, utility, and interoperability of research. Anita, we are looking forward to your talk.
Thank you so much for that wonderful introduction. And I think everyone can see my slide, yes?
Yes, it's fine.
Yes.
Perfect. So yes, I will be talking about how the literature has changed over the last 10 years and a little bit longer in a bit. And of course, the focus here is about research reproducibility. So there it is. Okay. So what about this reproducibility crisis? And here I just put a question mark. I'm sure that you have all heard about this reproducibility crisis, especially from your colleagues at the BIH. And so I don't want to hit you over the head with this too much, because I'm certain that many of you have already seen this figure from Leonard Friedman's work, and this figure from Malcolm McLeod's work. But effectively, I like this figure because what it does, whether or not the exact numbers here are correct, it does try to put a scale on the reproducibility issues that we are facing in science, and it also breaks it down into these smaller chunks. And I think if we see these as smaller chunks, it might be a little bit easier to try and figure out how to address these kind of smaller parts of a bigger issue. And so I will also point out how we've worked to address things like the key resources here, which is the biggest part of the chunks that Dr. Freeman points out, and also this study design issues, which effectively has to do with Malcolm McLeod's work and the work of Story Landis and others from the NIH, really pointing out that effect sizes are too large when certain things like randomization, blinding, and power analysis are not done by the study authors, driving these larger effect sizes that are not actually accurate. Okay, so looking now at how would we solve the research reagent problem, and how, is there even a research reagent problem? And for this, I like to pull out a paper. This one's relatively recent, but I could have pulled out just as easily a 2022 paper This still happens, in fact, quite frequently and a little bit too frequently. This is a paper recently published. Again, we're not picking on these authors. This is a kind of thing that happens a lot. And what they're saying is that this, you know, in their methods section, that this was blocked and labeled with mouse anti-flag from Sigma. And so if we go to then Sigma, and actually it's millipore Sigma now, we find 17 matches for this antibody. And so obviously this paper is not reproducible, and it's not reproducible for a really, really dumb reason. We know that these people know which antibody they used, but when they write about it in the paper, they don't give that information to the readers of the paper. And so if somebody is trying to reproduce this, they would effectively have to contact the authors. And that's bad, right? So what is a way for us to actually change this? And so we had talked to our society, the Society for Neuroscience, at great length to see if we could have this addressed as an issue. And we, and actually mostly the members of the NIH, had brought together a group of us, 25 journal editors-in-chiefs, as well as some informaticians. I was one of them, and others from NIH who had effectively asked the question of the journal editors, could we start to address this issue of bad reporting, especially of antibodies in the scientific literature? And over a series of 3 meetings, what we had agreed upon is that there would be a list created of all of the research resources that the Neuroscience Information Framework had already aggregated. We had changed a little bit about how you know, those things were displayed, including making sure that there's a persistent unique identifier called the RRID for each of these resources. So each rat, each fly, each antibody was given a unique number from the database that aggregated that information. And the full syntax was agreed upon by the journals to include the company name, which everybody used anyway, the catalog number, which most people did not use at the time, and also the persistent identifier, because sometimes companies either go out of business or they change their catalog so that this is no longer available as a piece of information on the web. So that's where really the persistent unique identifier comes in, because this record is not deleted from the authoritative database, and so you can always look up based on this number whatever happened to a particular antibody. Okay, so effectively the informaticians came together, and we just sort of made a long list. All of these little you know, cute icons, all they mean is that, you know, the rat genome database has a list of rats, and we pulled this list into a very long list of all of the kinds of reagents and resources that someone might use in their paper. And of course, that list is not complete. So for example, when somebody makes a new antibody, we say, hey, please go register that with the antibody registry. And then we put that into a single web portal, the RRID portal, which also has on top of that a way to search this list, right? But the cool thing about these RRIDs, these individual numbers for each of the reagents, is that you can then attach things to them. So not only is there information about this from Thermo Fisher, but maybe someone has used this antibody in a paper, which would then help their colleagues track down whether or not you know, this antibody would work in their application. So that's this portion of the project. And there are other people who say things about these antibodies or cell lines, such as the ENCODE project, the ICLAC, the International Cell Line Authentication Committee, says things about specific cell lines. ENCODE says things about specific antibodies, whether or not they are valid under particular circumstances and things like this. And so these numbers actually allow us to aggregate information around the reagents. Okay, So in 2014, we actually started the project with these 25 journals, and here is the result of the paper that we wrote about this in 2015, which shows that in the issue of the journal before we started the project, before we started asking authors for RRIDs, about half the time we could not find out which antibody was being used. But once we started asking for RRIDs, about 90% of the time, we were able to find the antibody that was being used in that particular paper. And this is also true for organisms. It was also true for tools, maybe not quite as big of an effect as with antibodies, but certainly this was an important finding. And this really encouraged more journals to come on board. And then a few years later, we had actually brought on board the cell lines. So from Cellosaurus. Cellosaurus is an XPASI project, which is a very comprehensive list of cell lines. And of course, here's a demonstration of one of these records where someone would type in their search term, in this case, 15C4. This is a cell line, and this cell line is known to be contaminated at the source. So this is one of those little bits of information that comes from the ICLAC, the International Cell Line Authentication Committee, which is a group of researchers who really deeply care about whether or not the reagent, the cell line, is authentic or whether it's not authentic. In this particular case, this is probably one of the worst examples where the cell line is contaminated, but often there are other things reported that allow people to use the cell line, but for example, a misidentification has been reported. And for those of you who might not realize this, cell lines are often, they come from patients. Patients are often misdiagnosed, and then that misdiagnosis goes with that reagent now throughout the scientific literature, and we later find out that if in fact a particular cell line is based on a hepatocarcinoma instead of a hepatocytoma, and so when we're talking about these things, we may not actually have the right cancer because it was originally a misdiagnosis. But what we did here in this study is we actually looked at the papers that were using cell lines, and we used some text mining technology to be able to take out everything in PubMed Central that was accessible to text mining, and we found 150,000 papers that had used at least one cell line. And then what we did is we did a match of these cell line names to this known list of cell lines that have a note about them like this. Not, again, all the notes were kind of pooled into one. So, and we found that 16% of the scientific literature out of this 150,000 papers were using a cell line that had a note associated with it. When we looked at the 634 papers where authors had gone into the portal, found the RRID, and then published that paper, it was only 5%. So the drop here in the usage of these potentially problematic reagents was 66%. This is, again, very highly significant results. So we think that not only are RRIDs very good for just identifying the things that are being used, but also they're very, very good at being able to understand when a bad reagent is out there. Even at the end of the process of publishing the paper, we can still clean up the literature from reporting on these bad reagents. Okay. So how many RRIDs are there? I just pulled this yesterday from our listings. So currently, we have a list of papers here on the right-hand side. And in 2014, there were just over 250 papers published in the first year of activities. And then in the last year, it was a bit over 10,000 papers that were published with at least one RRID. And we're currently at the 2,000 mark for 2022. And the reason I put this lovely little German flag here is because we actually do love our German authors quite a bit. because it seems like when they are convinced, when you all are convinced that RRIDs are a good idea, then you do a lot of registration of new reagents, which we then interact with you on through some of the databases that we run. And so this is just a thank you to the wonderful German people who are and authors who have been registering diligently new reagents with us. Okay, and the list of journals. These are the top journals at the moment with Cell Reports. It's a really big journal that has now published 67,800 RRIDs themselves. So this is not 67,000 papers, this is 67,000 RRIDs. There are many RRIDs per one of these papers. But you see also very small journals like the Journal of Comparative Neurology, who has published now about 9,000 papers, or excuse me, 9,000 RRIDs. And even though they only publish a few hundred papers per year, they have been with us and asking authors and really leaning on authors to get RRIDs into the papers. And so they're actually here at the top of this list, along with these really large journals like eLife and Cell Reports. And of course, the Journal of Neuroscience, which is our big society journal. Okay. So then how are these, so what happens to the RRIDs? And so these authors here, Nichols et al., right, published a paper and they said, hey, here's my mouse. And this is the name of my mouse, and this is the RRID of my mouse. And here's even a reference for that mouse of how that was created. And we say, hey, that's very cool. So our curators actually run a robot, but this robot is also run automatically on top of the open access literature, and we extract that information. So here the robot, you can see, notes the DOI of this paper, and it notes the RRID. So now what it does is it makes this little bit of information that says, here's a paper that's using this mouse. And the page that we created on the in our systems that talk about this mouse actually has a list of the papers that have used it, including this Nichols et al. Paper, which is actually this one right here. And that is a very cool thing. So these are bits of information that get extracted out automatically. And then here's our curator who has validated this particular record. And then that data was moved to the page that describes the mouse, which then can be used by others to figure out, okay, I'm trying to look for a mouse. Is this a good one for my particular topic, right? Or my particular methods that I'm trying to deploy. And one thing that we have never been, that has never been a problem with any authors is we actually have hooked up a Twitter robot that simply thanks authors for using RRIDs. So we tend to like that, and then that gets aggregated into the altmetrics as part of the papers that use RRIDs. So this is just a little, excuse me, thank you. A thank you for the authors. Okay, so now moving on to the investigator bias problem. Remember, we know that Leonard Friedman said that this was kind of the second largest issue with reproducibility. So there are plenty of standards, including the National Institutes of Health policy on rigor and reproducibility based on the Landis criteria. There is, of course, you are probably much more familiar with CONSORT or ARRIVE guidelines, which are endorsed by many, many, many journals. The more recent MDAR checklist, which is now employed at the at Science, at AAA Science. It's required there, and it's supposed to be endorsed, and it is endorsed by many of the other big publishers, and it's supposed to be moving to replace some of the other checklists that are reported. So there are plenty of checklists actually to and initiatives actually, like the UK Reproducibility Network and others, to improve the standards here. But we thought maybe we could use some of our prowess in AI in order to actually develop some algorithms to check the various rigor criteria. And so we actually developed 55 different algorithms, and that's increasing to search for various rigor criteria in the various guidelines. So here are just some examples of sentences that, for example, meet the criteria of consent. So written informed consent was obtained from parents from all participating children, and oral assent was obtained from the 7-year-olds, blah, 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 right? So this is a sentence that one might use, and this is a sentence that also would be required by the MDAR and the consort guidelines. So when we looked actually at the different types of guidelines here, these are the rigor adherence guidelines. One of the things you could see, and looking again at the MDAR checklist, ARRIVE, consort, the veterinary guidelines, as well as the NIH guidelines, certain things are required by nearly all of the guidelines, including things like randomization, blinding, power analysis, sex as a biological variable, inclusion-exclusion criteria, and attrition. Other items are required by fewer of the guidelines, but we thought, again, almost all of these are very important, and or all of these are very important in different ways. And so we actually developed those algorithms. So what we did in order to spread that to the community is we actually founded a company, and we asked that the journals actually start to use this tool, these algorithms, in order to publish their papers. So this is roughly what that process looks like. This is the Journal of Neurophysiology, where authors are asked not to go to our tool, but in fact they are asked to copy and paste a section of their manuscript into a little box within the author environment. So when you submit the journal to some journals, then you're asked to put your method section into a little box. And sometimes that says to send to SciScore, sometimes it does not say that, but that's fine. But that method section gets then sent to an API. This is an application programming interface. It's our tool. And the various things that we do happen to this particular bit of text. And what the tool does is it produces 3 reports. Most of the journals are looking at the core report, so which is the most complete. But I'd like to point out that if anyone is submitting to science, they will be asked to fill out an MDAR report. The tool, SciScore.com, will actually create that for you for free in about 1 to 2 minutes. And that way it's also verified by the tool. So you can get that and submit that to them for free, because that goes with the paper. Okay, so what does one of these reports actually look like? So it's broken up into 3 different tables. So there's the rigor table, so with all the rigor items. There's a key reagent table, which has all of the reagent items. And then there's an other entities table with things like statistical tests and oligonucleotides and any errors in RRIDs that have been noted. So looking a little bit more closely, there is a sentence here. So the tool is expecting something called inclusion-exclusion criteria, and then the tool finds a sentence from the authors that matches this particular criteria, right? So in the text, this is a sentence, and this would be something that it would say, hey, I found this sentence. And there might be things that are missing. So for example, in this paper, there is no attrition meeting criteria or a sentence that meets this. And so the tool reports that this has been not detected. The tool can also report that a particular entity, a particular type of criterion could be not required. So that way this does not go into the score. This does count against you in the score. This does not count against the paper in the score. In this section, there's also information about protocols. So in this case, the tool found a clinical trials protocol ID, which is this NCT number. And one of our journal partners had requested that we find the registration date and the start date of the study. And so we include a little warning about that when, the study registration date, which in this case was in 2020, was after the study start date, which in this case was 2012. For code information, the tool also finds either sentences about sharing code, or it finds the identifiers such as GitHub. And so in this sentence, the code found the URL for GitHub, and then the tool goes to GitHub and says, hey, do you exist? And in this case, this URL does exist. In this case, the GitHub repository is either private or, there has been some typo in this URL. So there's a nice check for that. The same is true for data information. This is a geo-identifier that does not exist. This is according to our tool. And this is a geo-identifier that exists. So people can go ahead and check that and verify that their identifiers are correct. With the RRIDs, with the reagents, we can have several different kinds of use cases. So in this case, the tool found several antibodies in this sentence, but found no RRIDs, so this would count against the authors. In this case, there is a mention of a database. So databases are one of the things that has RRIDs, and the tool is able to suggest an RRID to the authors of this particular tool. The authors did not put that in, but there's a suggestion to help them out. So this counts as sort of, partial credit. And then here the authors actually used an RRID for a common software tool. And that is, again, verified here. It's blue. Of course, in either case, the authors should go and find from the link that this is actually the appropriate RRID. The suggestion could be wrong in all cases. For RRIDs that have a problematic warning or a problematic or misidentified warning, those warnings are brought in here just as a little red dot, just to make sure that people understand that this there has been some issues reported around some of these RRIDs. And working with Dr. Weisselgerber's group, we are going to enhance this table of statistical tests to improve how much reporting we actually get from these sentences having to do with statistics. And of course, at the end of the day, we look for things that are there, and we look for things that we expect. And if everything that we expect is present in the paper, then the paper would score a 10. And if nothing is present, then it would score a 0 or rather a 1. And we basically round to the nearest number, and each paper is given a number. So you might ask the question, and I know that this is really small, but there are a lot of scores here. And the way that this could be read is looking at, for example, let's look at the consent. So here's a consent statement. We gave the algorithm 373 positive example sentences of consent out of a corpus of 78,000 sentences. And when we randomized this over multiple trials, the tool was able to detect about 94.75% of these things accurately, with a precision of 96% and a recall of 93%. Now that's one of our kind of better examples, but you can see that there are a lot of examples. There are some in the 90s, some in the this one's in the 60s. The power analysis is not very good here, detection of that, but we are certainly working on these sorts of classifiers in order to get better at some of this. And again, for some entities such as antibodies, we've already given the code 16,000 sentences. And so we're probably not going to do a lot better than getting about 80% of these, but still, this is much better than getting none. Okay, so we have a bunch of scores. Now we have a number for a paper, and or we can create a number for a paper. One of the things we can certainly do with that is to intervene with authors and ask them to fix, you know, typos and other things. And however, we can also do some fun things with these scores. So what are the fun things that we've done with these scores? There's a project that I think many of you have probably heard of, the Cancer Reproducibility Project. This originated from the Bagley and Ellis paper in 2012 that was published in Nature. Unfortunately, out of Amgen, a company. Unfortunately, the Amgen paper was not open source, and we didn't know really which papers were even being replicated or not being replicated in that, as the case may be. And so the Cancer Reproducibility Project, which is a big open source project led by the Center for Open Science, had been commissioned in order to try and replicate 53 papers that were the key studies in cancer. And Arrington just published the kind of summary after publishing many of the replication papers. And what they found was, in fact, you know, in terms of the original papers. So replication effect sizes were on average 85% smaller than the original findings. So really going along with what Malcolm McLeod's lab had shown earlier with a different field. 70% of the papers required asking authors about the identity or for the key reagents themselves. And out of those 69% were actually provided with key reagents or information about how to obtain them from the authors. So the authors were reasonably good at being able to, you know, after they were asked, but almost every, you know, a really large chunk of the papers simply were not reproducible as is. There was not enough information actually in any of the papers, so they had to reach out to the authors about the methods 100% of the time, and 70% of that time was about reagents. So another interesting part out of all of this was the original positive results were about half as likely to reproduce successfully than original null results. So actually, you know, if someone reports that there was no effect, they're right about 80% of the time, at least in cancer biology. But if they report a positive effect, they're right about 40% of the time, which is kind of interesting. So we looked in our recent, this is a recent preprint, so this is not yet published. We looked at the original paper's SciScores, and we asked, you know, what is the number, and is the number any different between any of these? So if we look at the original paper, and all of the papers that, again, had methodological issues were centering on a mode of a score of 3. And this is the spread. There was 1, 6, and one was a 0, one was a 1, but most of these averaged a 3. And when we look at the replication papers, the mode here was 8, or excuse me, the median was an 8 and the median was a 3, sorry, apologies. And so the replication papers whose goal it was to be reproduced further had scored much higher. They weren't anywhere in the 3 range. They were around the 7 to 8 kind of, and there was even a 10 in there, which is great. Okay, so we know that there seems to be at least this difference between the original papers, which you know, we can say very clearly that, you know, did not have sufficient information, and then these replication papers, which should have sufficient information to actually be replicated, and that difference is fairly stark. But what does the literature look like? So in this study, and again, this is an update to our original paper on the Rigor and Transparency Index, but now also addressing some. Looks like someone could mute potentially. Okay, thank you. So what happened here is that we looked at the PubMed Central open access subset. So this is about 2.5 million papers, manuscripts inside of the open access literature. We obtained scores, so there were method sections that were accessible from about 2.3 million of these manuscripts, and we ran these. This is coming all from about 9,300 journals, and we ran these with their SciScore algorithm. This actually the quote-unquote wall time, which means how long did the servers actually do their churning. That happened for about 2 months. And it produced for us a large corpus of data about each of the kinds of things that we look for, including, for example, statements about power analysis, statements about randomization, blinding, sex as a biological variable, and of course this average score, the SciScore for all of the papers altogether. And what you can see is that the average SciScore in the 90s, in the late 90s, was less than a 3 out of 10. And in the last couple of years, this was over 4 out of 10, which is about a 2-point difference here, or 1.5-point difference. But we still probably have a good ways to go in terms of making our papers better. So I promised to talk about the last 10 years, but this is a little bit more. But over the 10 years, the things that have been improving are randomization, although this isn't maybe as much as we would like. There's more of a steep curve here to improve randomization in the 90s to 2010 compared to this. But it still looks like it is improving. Sex as a biological variable looks like it's no longer improving in terms of being addressed. But actually power analysis is improving, although again with that one, this is only 10%. This is the 10% of papers line. So this and blinding can really certainly do much, much better. Okay. What about antibodies? We did focus a lot on antibodies early on, and so we looked at the antibody as a reagent from 97 to 2019, and this analysis again went over the 316,000 different antibody records, and what we were looking at here is what is the percentage of those records where our tool could actually detect a catalog number associated with one of those antibody entities. And it was around 10–15% in the 90s. And that has been shifting fairly quickly, especially recently. This has been a big shift. And I'm just pointing out here that this 2014 is where we started the RRID initiative. And in 2016 is really where some of the big journals, including eLife and Cell, had actually joined our efforts. And so they are driving increases in findability of this reagent type that are very stark. And because they're very big journals, they're actually starting to pull up the average of all papers. Whereas in some of the papers, like here in +1, where we are actually in the instructions to authors, but it's not very well enforced, and in OncoTarget, where there is no mention of RRIDs anywhere in the instructions to authors, those journals are actually lagging behind. Whereas these journals that enforce the standard actually are have very high levels of findability. Okay, and of course we like to look at Nature because everyone knows this journal, and so And they've also done a very nice job in terms of implementing this big checklist for reproducibility. And so we were looking, and this is again published last year. We were looking at the average SciScore of these papers, and here are the individual kind of pieces that go into that SciScore. So if you look at something like, whether or not sex as a biological variable is addressed in the papers in the 2010-2012 timeline, it's generally not addressed in more than 20% of the cases. And then after the implementation of the checklist, we find that now sex is being addressed in about 50% of the papers. Randomization of subjects into groups really did a nice jump here after the implementation of the checklist. And the SciScore itself, you can see that this is about 2.5, which, by the way, was severely lagging the rest of the journals, which were, about 3.5 here at the same time. And now Nature is higher than the average journal, so the average journal would be down here. So the average journals actually did not rise as fast as this particular journal for this checklist, but Nature is now leading reproducibility, but it was kind of lagging beforehand. So that is what that sort of looks like at the moment. And some of you may ask, because impact factor is such an important number for many people in the sciences, whether or not there's any association with impact factor. And the answer seems to be absolutely not. There is no relationship between the journal impact factor and the SciScore of those journals. And this is just the log scale of impact factor. And here is the impact factor as a function of quartiles. And you can see that there are dots, and each dot is a journal. So here's Pure J, and Oncology Letters is here. So these journals are all over the place. There is no relationship. Okay, but when we intervene with the authors, because I did tell you that we were trying to intervene with authors in some of these, with some of the society journals at least, and so we started the first journal that we actually started to work with was the Journal of Pharmacology. And so that's the line here in green. And in 2014, they decided, the British Journal of Pharmacology decided that they really want to do something about reproducibility. You could see here in 2014, they were actually behind other journals, and they had brought on their own checklist, which remarkably enough covered many of the same things that Arrive and Consort and MDAR checklist covers. They started to send this out to authors, and they started to get an increase in the SciScore here. And then they started a pilot with us, where actually I had actually become the methods editor for the British Journal of Pharmacology, and I was running the first version of the tool, which was not yet very pretty, and providing feedback to these authors. And so we got about a 1.5 to 2 point increase over 1 year, which was great. And now the journal is running the tool themselves, and they continue to see gains. It's not as fast anymore, but it is still continuing to gain in score. So authors are definitely seeing, or we're seeing many more of these criteria being filled by the British Journal of Pharmacology authors. And as a control, Frontiers in Pharmacology has not really changed very much. They change a little bit, but kind of along with the other, with all their other journal colleagues. So this is really an effect that can be measured. When we looked at the first 5,000 papers that had gone through the American Association for Cancer Research, this is just an average, daily average SciScore of anything that was run in this particular day over the first 3 months. of our activities with them, what you can see is that there is a nice trend that moves upwards. But again, these are, you know, initial submissions. These are final submissions. We don't make any kind of a distinction. And in fact, when we did look at the initial submissions versus the various revisions, we actually see a pretty nice trend where the authors are fixing something, and whether that's due to our tool or whether that's due to review, we don't know for sure. But we do know that the papers are getting better as people interact with our tool and as people interact with the reviewers. So something is improving here. And again, this slide is a courtesy of Daniel Ivanko, who is who works for the American Association for Cancer Research and has really been instrumental in implementing rigor metrics inside of that society. So here is something that Daniel actually also came up with. It turns out RRIDs are really simple to verify, because all you have to do in order to find them is type in RRID into a search system. And so he was able to show that, you know, when you just put in the instructions to authors, compliance with RRIDs is very low. But as you start to kind of ratchet up and use things like SciScore, you're starting to get higher and higher compliance. And in 2021, AACR has now added a statement where if the paper scores less than a 4 SciScore, then it will not be accepted into the journals. So effectively, we're yet to kind of see where that leads, but we are certainly getting some interesting results from that. Okay, so we also are able to, you know, again, divide up the information, these scores, these numbers into various ways. We can look at it as a function of country. We can look at it as a function of institutions. And so we were able to kind of see who has the highest, which institution has the highest scores. And it turns out the Oulu University Hospital actually takes the cake. Currently, they're averaging about a 5. And when we looked at German institutions, the Charité here is number, let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9. You guys are the 9th highest SciScore for German institutions. And this is being counted as the number of papers that we scored for this was just over 3,000. So blinding rates for all of those papers are 16%, which is actually relatively high. And randomization is being done at about 31% of the papers, and so on and so forth. So we can actually pull this out. But looking at different institutions is maybe not the only way to compare. Some institutions maybe do more physical sciences. Some institutions maybe do more things that we measure within biological sciences. So is there a way to maybe, you know, do a little bit more comparison in terms of like for like? And so my student, Peter, has actually created this algorithm where he looks at the title and the abstract of all of the papers that we're also scoring. He associates the affiliations, whatever people put into their affiliation string, with the ROR identifier for an institution, and then he looks individually at the list of affiliation strings, and he's trying to figure out which papers are actually similar, as opposed to what's the name of the department. Again, it's not the only way to score this, but it might be a little bit, might hopefully get some people thinking. So when we did this clustering for the University of California, where I'm from, we found that the University of California, San Diego Department of Surgery is actually most related to the UC Davis Department of Nutrition and the UC San Francisco Department of Pediatrics. Again, you would probably not think of putting these 3 departments together into a cluster. If it was me, I certainly wouldn't. But this clustering algorithm did actually come up with these 3 departments. But when we looked at the papers in those, in this cluster, they are actually, this is the most central paper of this, and you can see that actually this is about inflammation and serum zinc concentrations as related to nutritional determinants of anemia, which is a childhood disease. So it sort of makes sense that all of these departments are actually linked together, because it looks like they're all looking at largely childhood diseases. They're looking for a topic of paper. And so we have some confidence that this is actually an okay place to, an okay way to kind of look at papers or look at departments that are maybe lexically very different, like nutrition and surgery, but in fact, what they publish is relatively similar. So we did this analysis for different Charité departments to see how closely they are related to one another. And I do have to report that the Institute of Microbiology is quite literally out in left field in this particular analysis. Again, this is a multidimensional space that's being flattened onto 2 dimensions. It's possible that if you twist this a little bit, then this won't be quite so far in left field. But the rest of these clusters are, again, lexically potentially very different, but they do seem to form some clusters, like the departments of psychiatry here, and then an Institute of Occupational Health is actually being clustered together with these more psychiatry-focused things. And you can see, I looked at some of the affiliations from the people who have signed up early to this course, and so I just put little stars where I thought that we might have some people in the audience. But again, this is one way of looking at these departments and seeing how they are clustering together.
5 minutes to go, and maybe there's some questions. Can you try to
Yes.
wrap it up? I'm not sure how many slides there are left. So.
Yeah, I went a little slower than I needed to, so I think we can probably wrap up here. I will not talk about the screen at work because that is actually bringing in some of the our tools together with the tools from Quest and other places to address preprints. But I can wrap up here and stop sharing.
Thank you very much. Lots of material and also a lot of things to discuss. And thanks for also this little focus on BIH Charité. There is already one question in the chat. I'll read it in a second, but just to all of you, if you have questions to Anita, please type it in and I will relate it to her. The first question is by Christoph Stein, and it's an obvious one. It's about reporting versus practice, and his question is, how do you verify authors' statements, for example, blinding?
So blinding is one of the things that the tool picks up relatively well. Now, what the tool does not do is it doesn't say this blinding statement is appropriate for a study. But what the tool does say is we can find a sentence about blinding in this text. It's a very different kind of a thing. The authors could have said blinding is not appropriate for this study, and that would come for a point in that sense. Or they could say something completely ridiculous about blinding, but the tool only says, hey, the authors did address blinding. So in terms of how that works, the way that it actually works is simply that it's addressed. It's not that it's appropriate. I think AI is not quite there yet.
And not only appropriateness, but also the question, is it just a tick box exercise? I think that's where the question goes, like, could the approval that you have seen with all the checklists been one where people are now more aware of it, that they have to check these boxes and check them? But I guess you cannot look into this with your algorithm.
No, it's the algorithms are not yet good enough for that part.
There's another question, and that's by Carsten Krötzinger, and the question is, can researchers make use of the SciScore algorithm? Could I use it, for example, now?
Yes, you can use it. You can use it right now. I would recommend going to sciscore.com, and you should get 10 runs for free if you log in with your ORCID account. So if you log in, click on log in via ORCID, then we know you're a researcher, and you can then take your methods section. You can run it through the tool yourself. And see how well it does on your paper.
So please, if we have a little bit of time, so if you have questions, type it in. Here's one by Yatin Shah, and what are other use cases in the pipeline? Aha, what do you have?
Yeah. So I started to mention the use case for improving the reporting out on the kinds of things we find about statistical tests with Dr. Weisgerber's group, who have published on this kind of extensively. We're now able to detect some differences in what is being reported in terms of statistics on some of these studies. And we would like to give people a better contextual help to add the appropriate things to their statistics. So that is one thing. And of course, we're starting to really look more at the preprint literature, because preprints are a place where people, these aren't final products yet, right? So people expect review, and we can do some of this review using automated tools. We hope to make those reviews as useful to authors as possible, so they can fix the little things that they need to do to fix them, in order to at least make the reporting of the study much better. Things like looking up catalog numbers and persistent identifiers for their antibodies would be a relatively simple thing that would improve the paper.
Okay, thank you very much. I happen to know that there's another little thing in the make which has to do with your fellowship at the BIH, but that's probably something that will be reported at another time point. So thank you very much for your presentation. I'm sure that Anita would be available for questions via email if you have them. And as I said, she is around if you wish, even if it's far away.
Thank you so much. And yes, I'll be around all summer.
Okay. So my duty now is to also announce the next lecture, which is already in 1 week, which is kind of unusual. But it will be at the usual time. So it is, as always, at 12 o'clock. And we will hear from the editor at Cell. And this will be an insider advice from a scientific editor with all the secrets how you get to publish in Cell. So we're looking forward to that. And thanks for joining us.